So on this giant piece of paper, we are going to draw step by step the parts of mitosis, and we're going to discuss the major events that happen in those steps. So we're going to start in this spot right here, interphase, and we're going to take and just put a red star next to this, just for the purposes of saying we're starting here. It's a cycle. You could really kind of make the choice of saying I'm going to start in different spots, but it kind of makes the most sense if we start at interphase. Now, interphase, is that most of the cell's life or only a little bit of the cell's life? Most of it. About how much of the cell's life is in interphase? We've said about 96. I've seen some other literature that say 90, I've seen some that says 70, whatever. Most of it, right? The vast majority, 70 is way bigger than 30, 96 is way bigger than 4. Look at me, I'm adding to 100 in my head. Nice job, Mr. Babies. Thank you. Thank you. It's a tremendous Tuesday, that's why I can do it.
Tremendous.
On tremendous Tuesdays, adding to 100 is sometimes something I can do. So interphase is where we start. Now what happens to a cell during interphase? This cell grows. What part of interphase is growing? G1 and G2, yeah, right? Because G stands for gap, or we could pretend that it says grow. It really says gap, but G for grow, right? So the cell grows. So let's just pretend for a moment that that's G1. Because then after G1, we could talk about what comes next. And what comes next after G1, we get synthesis, or S phase. And what happens in S phase? It replicates. It makes a copy of the DNA, right? So we're going to copy the DNA. And at some point, we got to copy the organelles. Whether that's in G1 or G2, that's where we've talked about. I've seen both in literature. Okay, but at some point, we got to copy organelles. And then in G2, what happens in G2? Grows a little bit more. But most of the cell's life is spent in interphase. So what does a muscle cell do during interphase?
Grows.
Okay.
Appears there.
What about a nerve cell? What does a nerve cell do during interphase?
Senses.
What's that? No, the cell itself isn't going to replicate during interphase. What's that?
It's going to repair itself.
If it's damaged, it might be able to repair itself. Nerve cells aren't a great example. Okay, it's a poorly worded question, I think, because no one's getting what I'm trying to say.
Function.
Day-to-day function. Oh,
I
yeah.
literally just said that.
Okay.
Yeah, but
You ignore me.
Yeah,
Okay.
that's possible. Okay, so a nerve cell does whatever nerve cells do. A bone cell does bone cell stuff. A muscle cell does muscle cell stuff. So we can just kind of say daily, day-to-day functions. Okay, whatever day-to-day functions are. Muscle cell stuff, nerve cell stuff, that kind of thing. That's interphase. And at some point it's going to prepare for the division process that we collectively refer to as M phase. Okay? So, let's talk DNA. In interphase, what form is DNA in? Is it in the chromatin form, or is it in the chromatid form?
Chromatin.
Chromatin form. So it's
Uncoiled.
uncoiled.
It looks like spaghetti.
Okay? So it's like a bowl of spaghetti. Well, what part of the cell is our bowl? Where is the spaghetti contained within in our cell? The nucleus. Now, we are going to really oversize our nucleus. When we draw our nucleus, we're going to take up like 95% of the cell as a nucleus. I made that number up. Big. It's going to be like almost the entire structure is nucleus. Realize, not to scale, but per our model, where we care about the nucleus, and the genetic information in the nucleus, that's why we emphasize that. Are there mitochondria? Absolutely. Are there endoplasmic reticulum? Absolutely. Are there ribosomes? Yeah. But we're talking about the division of the genetic information housed in the nucleus. Therefore,
All I hear is
we're going to emphasize the nucleus. So we should make sure we label said nucleus, because otherwise it's just a circle on my piece of paper. So we want to make sure we draw a nucleus. Big circle, nucleus.
oh my God.
We've already talked about chromatin being uncoiled like a bowl of spaghetti.
Spaghetti.
So we are going to draw
Spaghetti.
our bowl of spaghetti in our nucleus. So using our red and our colored pencils, whatever colors you chose, we're going to draw a bowl of spaghetti in both the two colors. So red, and we are using red and blue.
Hi, spaghetti.
And hopefully that will show up a little bit better. But it's a bowl of spaghetti. Noodles going every which way.
Bless you.
Okay?
My noodles are important.
And that's how a cell spends most of its life, in that bowl of spaghetti, uncoiled chromatin form, 90-some% of its life. But then at some point, a cell has to move away from home, and it has to spread its wings and divide. I don't think that really fits in this analogy, but whatever. So then it goes to M phase.
And
Now, M stands for
mitosis.
mitosis, and if you recall yesterday, we talked about your new puppy called mitosis. And mitosis, your puppy, is at home, and your puppy is not quite potty trained yet,
So it uses a PMAT.
so we don't want him peeing all over our carpet, so we provided our puppy mitosis with a pee mat. So if it has to pee, it pees on the mat. Okay, so that is the order in which mitosis happens, and we can just go P, M, A, T. Now follow the arrows, right? We're P, M, A, T, right? P mat, and we're going to stop at that. Now,
Do they have to be capitalized or no?
yeah, absolutely, no, yes. We're going to capitalize them. Do they have to be? I don't know. I almost. Do you ever see prophase written in the lowercase? I think it's mostly capitalized. I don't know if it needs to be capitalized though. I don't know. I'm not sure. We're going to capitalize it.
For our sake, yes.
Okay. So P in PMAT stands for prophase. Now let's talk
Metaphase.
key functions. Okay. We talk about not key functions, key steps, key events that happen during prophase. So remember, the ultimate goal of mitosis is to create two genetically identical daughter cells. So we have to go through and we have to divide all of our genetic information. Now, what form of DNA what is the form that DNA is in during mitosis? Is it in the chromatin or the chromatid form when we pull them apart and when they divide?
Chromatid?
Chromatid, okay? So we have DNA in the chromatid. So at some point during prophase, we have chromatin. During the beginning of prophase, our chromatid tin condenses into chromatid, or another way we could word that is our chromatin becomes chromosomes. Because what a chromosome is it's the two sister chromatids being held together by centrioles. So chromosome is chromatid, but a lot of times we just say chromatin is in the chromosome form. So we have chromatin that is now in the chromosome form. Okay? The chromatin condenses and becomes chromosomes. At some point during this process, the nucleus has to dissolve. The nucleus disappears during the division. So basically our genetic information, our chromosomes are free floating, and the nucleus is no longer there. So we could say the nuclear envelope dissolves, or the nucleus dissolves, or the nuclear membrane dissolves. It breaks down. It's no longer there. So we can say that nucleus dissolves. And then there's these things called spindle fibers. And spindle fibers are like ropes, and I often think of them kind of like a lasso. And the idea of a lasso is I can take a rope, and I can throw it, and I can hook it onto something, like Mr. DeBoer, or whatever, and I can pull on it. Okay, and it provides a point which I can pull things around. Okay? And those spindle fibers are going to pull our chromosomes around. So those are the key steps that happen during P of our PMAT or prophase. So if this is what our nucleus looks like intact, what would be a good way that you think we could maybe show that it's dissolving or it's breaking down?
Showing it outside of
It doesn't
the nucleus.
leave. The nucleus doesn't leave the cell.
Well, we
the
could
nucleus
show
dissolves.
we could show it as dots.
Maybe we could show it as like a dot
Or
or dashed
dashes.
lines. Yep. So what we did have is we had a nice full circle, and what we can do is a nice way to kind of show that is maybe by putting some dashed lines, and then we'll add a little note that says the nucleus is dissolving, right? To show, or maybe, should we draw? Yeah, we should probably label the dashed line specifically that it shows, compared to drawing an arrow from our key points. That might get a little messy, right? Yeah. So our dashed lines is a way to represent that we have our nucleus dissolving. So our nucleus is dissolving, so we can do that by a dashed line. And now our bowl of spaghetti is no longer a bowl of spaghetti, it's a cracked bowl. Ooh, that didn't sound right. It's a broken, shattered bowl. There we go. That's a little better.
Not
It's a
a cracked
shattered bowl
Not a crack bowl.
of chromosomes. Okay, it's a shattered bowl, dashed lines, of chromosomes. The bowl is disappearing, and we're going to draw chromosomes. And we're going to draw a total of 4 chromosomes. We're going to draw 2 blue ones and 2 red ones. But we're going to be pretty specific with them. We're going to draw 2 big chromosomes. We're going to draw a homologous pair, large chromosomes. And homologous chromosomes, one of those homologous chromosomes came from your, and one of them came from your
Parental unit.
dad. So we're going to color code them, mom versus dad. So we're going to draw a big pair of homologous chromosomes, 1 blue and 1 red. And we're going to draw a homologous pair of chromosomes, little ones, 1 blue and 1 red.
I messed up. Well, that fixes it.
Okay? So these big chromosomes, homologous pairs, the 2 little chromosomes, homologous pairs, and you got one of them from your mama and one of them from your papa, or your mom and dad, or whatever you call them, from your parental units.
Parental unit.
And then we have these little dots that Mr. DeBoer is drawing, and those are referred to as centrioles. They're really just anchor points that spindle fibers start to grow out of. Sometimes we draw, sometimes we label the centrioles, sometimes we don't. I don't think we're going to today. But the centrioles are the things that an animal cell spindle fibers grow out of. But it's an anchor point, it's an attachment point. And these lassos, these ropes, these spindle fibers, it's like wrapping my lasso around the horn of my saddle, right? If you're gonna pull a cow on your horse with my lasso, boy, this is something that everyone can relate to on an everyday basis.
Yeah, definitely.
If you just pull on that cow bite with your hands, you're probably gonna fall off the horse. So you wrap it around the saddle horn, and then your horse can kind of help you pull. It's kind of like a centriole.
Yeah, because I know what that's like.
Yeah. I've watched Western movies.
Or you could have
No, I've tried doing this in
used
real life.
a dog with a leash example.
I suppose a leash would maybe be a little bit more relatable, but it's not what I chose to go with for some reason. So, our puppy, mitosis, sometimes needs to use a PMAT. So during the P phase, prophase, the nucleus dissolves, chromatin becomes chromosomes, and we start to form spindle fibers. That's prophase. PMAT, prophase, metaphase, 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 metaphase. There's another M word that I used a bunch of times when I talked about metaphase yesterday. Middle. Okay, so when we draw metaphase, we're going to start putting some things into the middle. Now, what would you anticipate goes to the middle if we're talking about dividing genetic information? The genetic information, right? So what form is that in? It's in the chromosome. So we're going to draw our chromosomes in the middle. So one of the big things before we draw it, we're going to talk about it. We're going to talk about the major events. Is the spindle fibers are going to. I don't know if I like how I have that worded, Mr. Board.
Mr. DeBoer,
Instead of spindle fibers attached to the centromere, should we say the spindle fibers
It's fine, it's fine.
pull on the chromosomes? You know? Maybe we go bullet point number 2 first. The chromosomes go to the middle. The chromosomes go to the middle. Meta, middle, middle, meta, meta, middle, 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 meta, meta, middle. MM. Chromosomes move to the middle. How do those chromosomes get to the middle? What pulls those things around in the cell? What is the lasso?
Spindle fibers.
The spindle fibers, right? So the chromosomes move to the middle, and the spindle fibers pull on the chromosomes, line up the chromosomes, move the chromosomes to the middle. What should we write, Mr. DeBoer? Chromosomes, spindle fibers.
Align. Spindle fibers.
Pull on.
Align the chromosome. That's
Were you okay?
Spindle fibers
Were you okay?
pull on, line. Let's just say pull on chromosomes. Okay. The chromosomes, the spindle fibers pull on the chromosomes, and the spindle fibers pull those chromosomes to the middle. Okay? I didn't love this centromere part, Mr. DeBoer, just because the kind of the conversation we were having about plant cells.
Yeah.
So the spindle fibers pull on the chromosomes, and the chromosomes move to the middle. Okay, now there's 2 different ways we could draw the middle. There's the equator version, or there's the prime meridian. I think that's the north-south one, right? Or the international date line. Well, we're going to go the equatorial, thank you, the equatorial version of this. So we are going to draw our chromosomes along the equator, because the way we have our bubbles splitting in early next phase is going to be better here. So we're going to put our 2 homologous chromosomes next to one another, but they wouldn't necessarily have to be. So we're going to put our 2 little chromosomes on the left side, and we're going to put our 2 on the right. Left versus right, it doesn't really matter, but the idea is we have 1 big red, 1 little red. 1 big blue, 1 little blue. These are homologous chromosomes. Now, those centrioles, those little kind of anchor points, are now going to be at the north and south pole. If we're talking about the equator, the north and south pole. Okay, they're going to go at opposite ends. That's the key thing here. And those spindle fibers, those rope-like structures, are going to be attached to the centromeres, and those are what are going to be tugging and pulling on those things. And they're playing a game of tug-of-war right now. And at this point during metaphase, they are lined up in the middle. No one is winning the game of tug-of-war. It's a tie. They're just kind of sitting in the middle, but they're still pulling. And that's what we get when we get to middle. Middle, middle, middle.
Do you have to label them or no?
In terms of things to label, there's not really a whole lot to label on this because the key points I'd say are kind of fairly descriptive.
I mean, if you want to relabel chromosomes and the spindle fibers, go ahead, but
Yeah.
that's up to you.
Okay, so our puppy's name is mitosis, and our puppy mitosis sometimes needs to use a PMAT, prophase, metaphase, anaphase, and anaphase is the 3rd phase of mitosis. And in anaphase, ana apart, apart, ana apart, apart, ana, ana apart, things are going to get pulled apart. Now what's going to get pulled apart? What object is going to get pulled apart here? Chromosomes, right? We're going to pull a chromosome apart. And when we split a chromosome apart, what is one half of a chromosome called? Yeah, they're sister chromatids, right? So what's going to happen during anaphase is we're going to pull the sister chromatids apart. Okay, the sister chromatids are pulled apart, or you could use they're pulled away, but we often talk about pulled apart. So we take an chromosome, we take our sister chromatid, and we rip one sister chromatid this way, and one gets ripped this way. The game of tug of war has taken a very violent turn. And instead of one team winning,
They both lose.
it gets ripped in half, and they both fall down.
And they go boom.
Okay? And they go boom. And the reason why these are getting pulled apart is because the spindle fibers are getting shorter. Okay? So those ropes, they're pulling, they're tightening those ropes, they're pulling on the ropes, and those spindle fibers are shortening. Because the spindle fibers are shortening, something's got to give. That protein, that centromere, is starting to dissolve, it's weakening, they get ripped in half. Now, when we draw our sister chromatids getting pulled apart, remember, if this is my chromosome, from here down this is my sister chromatid right here. This is my sister chromatid right here. This is my sister chromatid, this is my sister chromatid. Okay? So when we draw that, we still kind of keep our X shape, and we pull them like this. Okay? We're going to split our chromosomes like this. So here's Mr. DeBoer's big red chromosome. This is his big red chromosome that the sister chromatids got ripped apart. Okay? They got pulled apart. Here's Mr. DeBoer's little red chromosome. Here's Mr. DeBoer's little red chromosome that got pulled apart. And then we repeat the process for the blue chromosomes. The sister chromatids of the blues are ripped apart. And they're going to the north and south pole, if we were going to refer to this like as a globe. They're getting pulled apart. They're getting pulled towards the centrioles by the spindle fibers that are anchoring those things and are pulling on those things. Okay, so we have some spindle fibers that are pulling on those ropes. Our puppy mitosis needs a PMAT, prophase, metaphase, anaphase. Anaphase part. The spindle fibers pull and rip the sister chromatids apart. Our puppy needs a PMAT, prophase, metaphase, anaphase, T, telophase, sometimes pronounced telophase, but I say telophase. And when we talk about telophase, we often slash telophase and we combine it with cytokinesis. Now, cytokinesis and telophase are technically two different parts. Cytokinesis officially is not part of mitosis, but it's a pretty important step that has to occur during the cell division process. And it kind of occurs at the same time, so I often slash it and consider it to be kind of the same. They're occurring at the same time, but they're technically two different things. So remember, our end game, or our big goal, is we're going to end with two cells back in interphase. So how do we end with cells in interphase? Well, we got to put things back to the interphase form. And what form was the nucleus in during interphase? Was there a nucleus or no nucleus? In interphase.
There was.
There was, so we're going to have to put a nucleus back, right? What form was the DNA in during interphase? Uncoiled. So we got to uncoil the chromatids, right? So those are some kind of big things that happen. So the nucleus is going to reform. The chromosomes go back to chromatin, right? So we're uncoiling the DNA. That's one of the key things. We're not actually going to draw the uncoiling process yet. But it's what starts to happen. And what we start to get, and this is the cytokinesis piece, is we get this cell membrane that starts to pinch inwards. Okay, the cell membrane starts to pinch, and it kind of looks like, to me, it looks like a peanut. Like a peanut that has two of them in there. You start to kind of get that
See that?
pinch inwards.
Does it need to go higher? Can you see it okay?
A little bit higher.
Okay, so the nucleus reforms, the chromosome is going to chromatin form, the cell membrane pinches, and then in the process, we end up with our final thing is we finish with two genetically identical diploid cells. We finish with two genetically identical diploid cells. And we'll go through and we'll probably add another piece or two. Okay, so we add two genetically identical cells. And they're going to be diploids, but we'll add that at the next piece. So we're drawing it going back to the transitional phase, right? So we can draw the nucleus reforming. Now this is going to become a cell, and this is going to become a cell. So we can draw two nuclei starting to reform. It's the reverse. All right, so we drew a dashed line to show it's dissolving. Now we're drawing a dashed line to show that it's reforming. And then we have our chromosomes will start to go back to chromatin, but we're not going to draw it there yet for our illustration purposes. We are going to draw our sister chromatids on the bottom. So we had 1, 2, 3, 4 sister chromatids. We're going to draw 4 chromatids, but we're going to draw them as a stick. We're not going to draw them bent anymore. We're going to straighten them out. So we're going to draw one big red chromatid and one little red chromatid. We're going to draw one blue, big blue, and one little blue in our bottom cells. Because that little chromatid is that is that, right? And then what's true is it's the same up above because the sister chromatids fold apart. And hopefully very quickly here, we can finish up our colored pencils. And if we go to our last two bubbles, now this is going to become a cell, and this is going to become a cell. So just so we can be done with our colored pencils, what's going to happen to our DNA is going to be a bowl of spaghetti. So we need two bowls of spaghetti. We need a bowl of spaghetti here, and we need a bowl of spaghetti here, which means we need a nucleus. We need a nucleus, we need some spaghetti, and we need some spaghetti in the two colors. We'll label those final pieces tomorrow, because we're out of really quick here. Wait.
We get on regular time again.
Oh, I was thinking 06. Take a deep breath, Miss Davies. We have time. Gotcha.
Don't die on us.
I'll try.
Try not to die too hard.
Okay, so we have two bowls of spaghetti. We have some red spaghetti and we have some blue spaghetti. And we need to have our bowls to put our spaghetti in, which we call a nucleus. Okay? All right, so because I don't know how to read a schedule, we need to backtrack, and there's two things we should label on our other thing. Okay, so bowls of spaghetti. Okay?
I like spaghetti.
You have blue and red spaghetti? Okay, red spaghetti is pretty self-explanatory.
Okay, so we're going to go back to our telophase cytokinesis picture because there's two things we should label on our diagram. Two big things. This is what are we trying to show here?
Nucleus.
Our nucleus reforming. So we're going to label one of the dashed lines and say the nucleus is reforming because that's a big thing that happens in telophase. So we're going to label that and say, hey, look, the nucleus is reforming, and that's what that dashed dotted line is representing. And then we start to get this inward pinch of the cytoplasm. So we're going to talk about the cytoplasm as being pinched. Or pinches, or, I'm sorry, the cell membrane. I said cytoplasm. I meant cell membrane. The cell membrane is pinched inward, or is pinches. Pinched, maybe past tense? I don't know.
Pinch membrane.
Cell membrane.
You decide this one.
Pinch.
Pinches, yep.
Pinch.
So we get that cell membrane starting to pinch inwards. And if we pinch this hard enough, long enough, what happens is it comes through and then, boop, they split into the two new cells, which we drew over here. So the top half goes up here, the bottom half goes over here, and what we end up with is two cells. Now, what do we refer to those cells as? We start with a parent cell and we end up with daughter cells.
Oh, wait a minute.
Okay, so when we go through and we talk about these two cells, this is a daughter cell and this is a daughter cell. So we're going to say that we end up with two daughter cells. And what's true about those daughter cells?
They're identical.
Genetically identical to one another. They are genetically identical to one another. Maybe this is a good time to stop this recording. We'll finish these last few words that we need to tomorrow because we can give you some time for your color concepts. Great question. And the 11 by 17, when you fold it 17 in half, it's 8.5 by 11, which is a standard piece of paper, which will fit very nicely into your folder. So you want to fold these in half. I would fold these in half because they match a regular sized piece of paper, and then you can put them in your folder. Have a terrific Tuesday, tremendous Tuesday. Oh wait, yeah, you can't leave till the bell rings.
Yeah, don't do that.